Hi everyone, I'm Vedika and I'm pursuing my MTech in biotechnology from Amity University, and I am a research fellow at Pine Biotech. So I will be talking about my project today. The title of my project is Transcriptomic Analysis of COVID-19 patients and their clinical data. So to begin with, the coronaviruses are a highly diverse group of transmittable viruses that infect humans and animals alike and can cause mild to severe respiratory infection. The ongoing pandemic that we are seeing is the COVID-19 pandemic. It is caused by the novel beta coronavirus strain SARS-CoV-2. It is of zoonotic origin and possesses extraordinary threat to public health, safety, and progress. The disease can be classified into 3 to 4 stages, which are as follows. Number 1 is the asymptomatic stage, which includes the incubation period from 1 to 14 days. Number 2 is the mild disease, which is followed by symptoms like fatigue, fever, cough, etc., which can also progress to the moderate disease where some dyspnea and respiratory difficulty can be observed. It can also progress into severe stage in some of the cases where some medical attention and hospital admission may be required. Also, coming to the emergence and spread of the disease, in December 2019, according to the timeline, first 27 cases of a viral pneumonia were documented in a seafood market in Wuhan. In January 2020, the Chinese government identified the pathogen as a new coronavirus variant, and towards the later half of the month, WHO declared it as a pandemic PHAIC alert, and human transmission was confirmed. It is suggested that the virus is of natural origins and originated likely in humans. In the first quarter of 2020, the viral infections had also reached India. It has occurred in 3 waves so far. 3.6 crore cases have occurred, and the recovery rate remains close to 98.18%. To understand the structure of the virus, we would say that the virus has various proteins like spike protein, the envelope, and the membrane, followed by the presence of a nucleocapsid inside in which the single-stranded RNA is contained. It also contains a replicase enzyme ORF1A1B, which is a fused ORF. Thus, it has 6 functional ORFs, which go in the order of 5' to 3'. Also, several ORFs encoding accessory proteins were observed. Coming to the genome sequence identity analysis of the virus, it was showed that it shares maximum identity of up to 90% amongst all animals with bat virus. And coming to the viral taxonomy, it shares 79% identity with SARS-CoV and 50% with MERS-CoV. Also, initially, it is the spike protein of the virus that is responsible for its antigenicity and recognition by the ACE2, which is the angiotensin converting enzyme to receptors. The virus thus enters the alveolar epithelial cells in the lungs, and it is followed by replication and subsequent migration down to the airways, which can progress to the lungs and the respiratory organs. Thus, it causes respiratory distress and discomfort in patients. Thus, to analyze and develop therapeutic targets against the COVID-19 disease, it is very important that we understand the transcriptomic changes that are associated with the clinical progression of the disease. So with the same aim, a pipeline was developed and all the stages were carried out on the T-Bio server and various interactive and interesting pipelines were followed that gave very graphic results. The dataset number GSE 17867 was obtained from the GEO website. 144 samples had progressive data, which means that at day 0, they had either asymptomatic or mild disease. And at day 5, the disease had progressed either to the moderate or severe disease. The rest of the patients had only data for one of the days amongst day 0 and day 5, and thus they were excluded from the study. The Q score of all the sequenced genes and the NGS data, which was less than or equal to 5, were removed, and the sum of the gene expression was reduced, and those with N value greater than 10% were only included in the study. It was followed by the principal component analysis step. Here, first quantile normalization using the T-bio server was performed. The log transformation occurred and the threshold was kept to less than 5, followed by PCA. The differential gene expression analysis can also be carried out, which converts raw reads to statistically significant results. The log two-fold chain values of greater than 1 and less than -1 were only kept. Also, as we compared multiple genes, it was important to look at the p-adjusted values, and the FDR was kept to p less than 0.1, so that only significantly differentially expressed genes and significantly the downregulated and upregulated genes were selected. Thus, after all the filtering and sorting, 28,641 genes were taken for further analysis. Now the results were obtained, and we performed a principal component analysis, which is basically follows a dimensionality reductional analysis. As we have data across a lot of samples, and a lot of patients, and for different types of genes that are differentially expressed, they form the various components in PCA. Thus, it is important to reduce the PCA, the components, into lesser number of components through the formation of various eigenvector matrices and interpret the results as the same. We observed that a PC1 value of 21.47 was observed, PC2 value of 15.47 was observed, and a subsequent PC3, PC4, PC5 to PCN values were also observed, but the highest PC values were taken to be plotted amongst the x-axis and y-axis respectively. The blue dots reflect the plots in samples on day 0, and the red square dots depict the progressive samples on day 5. So as we can observe in the graph, the pattern is observed as such that the day 0 samples were observed in the second and the third quadrant, and the day 5 samples were observed in the first and the fourth quadrant in the graph. Thus, the PCA scatter plot says that significantly differentially expressed genes are observed in the mild and progressive stage patients, and the significant difference is observed on the PCA plot by the segregation of the plots in various quadrants. The immunological profiling of severe to moderate COVID-19 patients is characterized by increasing order of density of the regulation of pathways such as JAK-STAT pathways, cytokine-cytokine receptor interaction, and hematopoietic lineage, which leads to pronounced T-cell lymphopenia. Also, it is important that host antiviral responses are innate in nature and involve cytokines and interferon-induced proteins. The key differentially expressed genes that modulate the significant pathways in genes are identified to various degrees in moderate, mild, severe, and asymptomatic COVID-19 patients. All this can also lead to the development of a hyperinflammatory state. And when the viral load was correlated with the disease severity, it showed no significant correlation, which also states that even mild patients or patients that have asymptomatic and mild disease are highly infectious. Thus, it is important to have steps like social distancing and hygiene, even across all patients, and even non-infected people by wearing of masks, etc., to prevent the spread of the same. So to conclude with, I would like to say that the transcriptomic analysis showed that the transcriptome of SARS-CoV-2 infected cells reflected the interplay between host and virus, which can provide valuable insights into mechanisms underlying SARS-CoV infection development and the COVID-19 disease progression. The viral infection triggers a large-scale host immune response and leads to the upregulation and downregulation of hundreds and thousands of genes of increasing order of severity according to the types of pathways that are involved. Thank you so much.